our next speaker, who is Marijonas Tutkus, who is an associate professor at Vilnius University, who is now transitioning us from the meso-imaging scale to the super resolution level and telling us about a super resolution system for single molecule localization using the miEye system. Please go ahead with your presentation.
So it's a pleasure for me to present our work in this venue. And I would like to tell our story, how we got into here and what is our newest developments and achievements into this open source microscopy work that we do. So we call it miEye. Well, it's a microscopy eye, let's say, and it's a benchtop microscope. So since we are working in Lithuania, we don't have kind of unlimited funds. So we decided to build our own setup and to develop a software that we can trust, understand, and we can optimize. And the software that we are using for controlling the microscope is based on Python. Like most of the people are moving to that. We were working in the lab with protein DNA interactions, as you can see on the screen in vitro, and now we are transitioning into the living cell experiment. And for understanding protein DNA interactions or any other actually interactions in living cells, super resolution fluorescence microscopy is a really good tool. And so we had like 50,000 euros and we decided to build our microscope, which you can see on the bottom. And now I'm going to introduce you to the super resolution microscopy, and then I will explain how our developed system works and what cost-effective solutions we decided to take. So in this figure, you can see that with the super resolution microscopy, we can probe objects that are 10 nanometers and below. Now with the MinFlux, we can achieve also 1 nanometer and lower resolution, which is even better. But with a standard single molecule localization microscopy, we can work definitely at 10 nanometer resolution, and we can probe such objects as viruses and molecules. How does it work? So single molecule localization microscopy is relying on localizing single fluorophores. So in original fluorescence image, let's say here we have 8 filaments that are labeled with fluorophores. So you would see in normal fluorescence micrographs all of those dyes and fluorophores overlaying each other, and you wouldn't see individual molecules. While in super resolution single molecule localization microscopy, we can play several tricks. So we can make them blink or switch or bind reversibly to the targets. And once we can make them blink, we can identify their center positions. So normally their PSFs or point spread functions are in the order of 300 nanometers in width. But once we collect sufficient amount of photons, and the more photons we collect, the better we can tell where the center position of these PSFs are. So now the resolution scales in this method with number of photons. So everything what is related to the detection has to be optimized to the perfection, because more photons we collect, the better we can tell where the center position is. So if we play such a game, for example, if we can switch these fluorophores, now I'm showing you a cell, say a bacteria cell with multiple proteins labeled with a fluorophore, we can excite them, and then we can detect fluorescence. So if they are shining all together at the same time, that's not really beneficial. But if we can try to do the blinking trick, either by shining really high intensity laser power, like in a storm method, or by adding some environment changes that could start them blink, we can identify individual molecules in different frames. So we have to acquire a number of frames, let's say 20,000 frames, with a short exposure time, and identify fluorophores in these frames. So I think I just skipped one slide here. So basically, I would show here an example from different study. This is not my own study, but I'm just representing the data so that you could get a intuition of what's happening here. On the left-hand side, you can see microtubules in the regular image, fluorescence wide field micrograph. And then in the middle image, you can see super resolved microtubules with the storm method. And the right-hand side shows that the resolution is around 24 nanometers. This is the full width at half maximum of the PSF. Now the resolution is the way how we estimate it is slightly different. We would use FRC, Fourier ring correlation, but nonetheless, this is a good approximation. And in order to collect such images, we had to kind of record 40,000 images and then do 3 million localizations. So we can understand that data analysis is also tough here, and we have to speed it up a lot if we want to see the data visualized quickly after the experiment. And okay, basically we have different microscopes available on the market. There are commercial solutions like N-Storm from Nikon, or there is also an Oxford Nano Imager, a small box, a very nice piece of equipment that can allow you to basically do these experiments: Storm, Palm, Paint, other single molecule localization microscopies. But the price of these instruments is above 200,000 euros. So this is typically a lot of money actually for a small project. And people started to develop different projects. One of them is called miCube, developed by Hannes Hohelbein. There are also other cheaper solutions, and they are ranging somewhere from few thousand to 50,000 euros, depending on the equipment that they are using. So here we are going to our miEye microscope, and we have our microscope, which is a box of aluminum, CNC milled. We call it main body, and objective is standing on this CNC milled body. Then we have excitation paths on the right-hand side and the emission path on the left-hand side, and we have our own laser engine that is coupled through laser fibers through the optical fibers to our setup. And we have our miCroEye solution on the computer, obviously, which is controlling this microscope, collecting data, and we can visualize the data on the real time once it's collected. So this setup is published in HardwareX in 2022, and we also have our GitHub web page where you can find our miCroEye present. And one of the things that we have to kind of check where we can cut down the price is the lasers. So typically people will be using high power single mode laser boxes that cost about 50,000 euros or more. And we decided to actually use our Lithuanian colleagues' laser source, which is a multimode open space laser. When we couple it with multimode fiber or single mode fiber, and these two paths that you can see here are basically interchangeable. So that we can use single mode fiber for turf illumination, and we can use multimode fiber for epi type of illumination when we want to study thicker samples. And we use turf excitation when we want to study something that is close to the surface of the cover slip. And we interchange in between of these two excitation modes just by using a mirror on a magnetic beholder. So it's very easy to do that interchange even in a single experiment. So our laser is around 10,000 euros, and then the rest is just the fibers and coupling optics. Obviously, when we work with multimode fiber, that's good because we can couple the laser power very nice from the source up to 90%. But the drawback is that we get speckles all the time because of the mode mixing. So to decouple that, we are using agitation module. You can see that the orange fiber is rotated around a agitator motor on which is an aluminum box, and this agitator motor is just vibrating the fiber, so we can get the mode mixing, and then we avoid the speckles. As you can see here on the graphs, on top we have it illumination profile without agitation at different exposure times, and at the bottom we have illumination profile with the agitation motor on, and it's essentially flat irrespective with the exposure type. Then with the single mode fiber, as I said, we can work with epi, high-low, and turf modes. It all depends on the angle of incidence, and we expand the beam as much as we want. This is a kind of parameter we can change. And then the expansion of the beam is basically setting the flatness of the illumination field. But the drawback with the single mode fiber is that the coupling is not perfect, and we typically couple no more than 20% of the whole open source laser box into this one. And the last thing that we can cut the price down with is the CMOS camera. We are using industrial CMOS cameras instead of scientific CMOS cameras. because the industrial CMOS cameras with Sony IMX sensors, they cost around 600 euros instead of at least 15,000 euros of scientific camera. The drawback of the industrial CMOS cameras is that they heat up a lot during the normal experiments. So here on the right-hand side, in the top of the page, you can see the working temperature versus exposure time for different IMX sensors from first generation, second generation, and the fourth generation. And we decided to build our own cooling elements on these industrial CMOS cameras. So basically, we attach the TEC element and a water block on top of that to remove the heat from TEC element, and we can set the operational temperature of the industrial CMOS camera stably at 15 degrees Celsius almost, irrespective with the exposure time. With this, we can achieve the readout noise, which is in the table for the fourth generation camera, which is Sony IMX 547, very similar to the commercial Orca Fusion readout noise, and the dark currents also very similar. So this is something that we work now in the lab at the moment, and we are developing this technology, and I think we would greatly benefit from working with industrial CMOS cameras also, because they are having global shutters, and so that avoids the distortion possibly that we can obtain in the experiments. And the last piece of development that we've done is the miCoE Python toolkit. I'm showing here you the TIFF viewer. This is one of the three modalities or modules of our software. This allows us to do single molecule localization, basically real-time single molecule localization. And if you really like this, you can just download the Python toolkit and use it for your own purposes. We also have the microscope control module, which allows us to use to trigger lasers, set their powers, do the autofocusing, also whatever hardware is connected, filter, wheels, this can be controlled through this microEye control module. And the last modality is the data acquisition module, which works with multiple industrial CMOS cameras. And we can set the parameters, we can set the acquisition parameters, and we can store the data. And here I'm showing you results which we achieved and acquired using D-storm and nucleophorenes with a snap tag in UTOS cells that were fixed. So basically we fixated the cells. We labeled the nucleophorenes with snap, red snap fluorophore. And then we did blinking of these probes using high laser power and reducing conditions. You can see nicely resolved rings of nucleophorenes, and you can see each monomer out of 8 monomers that contains single rings. And the resolution, the ground truth resolution that we can achieve with this sample is around 20 nanometers, which is actually really good. So it means that the true resolution is even lower. We also can do the paint type of studies where we see imager strands binding and unbinding from the Docker strand, and we can detect them while they bound to the Docker strand. And we're using that obtained nano rulers where Docker strands are placed on DNA origami, which is having a shape of a ruler. And they are separated by 80 nanometers. So you can see on the left-hand side in the figure A that we can definitely resolve those 3 dots separately. They are spaced in 80 nanometers from each other. And the resolution that we can achieve with this sample, the FRC resolution, is around 18 nanometers. And here in the bottom, I'm showing you how results would look like if we have no cooling on the industrial CMOS camera. That would be these red dots. They are kind of making 3 clouds. You can still identify them on the ruler, but when you cool the industrial CMOS camera, they are resolved so much more nicely. And that just shows how important it is to cool down the camera and to have low noise to detect them reliably. We can also, so our last goal is to do the single molecule tracking inside of the limiting cells, because these are the most interesting experiments for us. And for these experiments, we are using a single particle storm tracking, meaning that while molecule is in the on state, we can track their positions, and we can do that for all the molecules, and they would be repeatably blinking. So it was reported previously by by Stefan Uhoff that we can achieve tracks up to 12 frames with halo tags using such a method in living bacteria cells. And this is actually really good compared to what we would achieve with photoactivable fluorescent proteins, because they bleach too quickly. We already tried that in our experiments, and we typically get tracks of 2 frames, which is not sufficient for our goal. So, but also very importantly is actually to say that the microscope has to be really stable. I didn't mention that, but I had these curves here that represents the lateral drift of the sample in X and Y in the middle of the top row. And over 20 minutes, we have a drift of around 30 nanometers laterally. And this is a really good result, I think, that we can achieve because of the CNC milled body of our microscope and also very stable motorized stage that we are using. So nonetheless, if we are probing movements of 8 nanometers, like in this case, kinesin is moving on the microtubules, the drift of the microscope could be a problem, right? So, and actually the microtubules and the kinesin is a good model or a standard for single particle tracking, because microtubules are like a straight lines on the surface if you mobilize them, or even in the cell, they are kind of more or less straight, or at least you know where they are. You can just label them if you want to. You can label your kinesin with the halo tag and do the labeling with the red fluorophore and do single particle tracking storm experiments. These proteins are moving in living cells like 1 micrometer per second, and their tracks are around 1.5 micrometer until they dissociate. So it's not that hard to prepare such a sample. And here you can see our data when we track movement of kinesin in living COS7 cells on the surface. They have microtubules labeled by GFP. You can see them overlaid with the tracks of kinesin, which was red labeled. And we did storm experiments. So the tracks that we obtained sometimes are overlaying with the tubulins. Sometimes you can see that the tracks are not really on tubules, but that could be also the reason, because not all of the microtubules are labeled. The ones that are endogenously expressed, they would be without label. And now we are working on how to label all of them, basically, with our labeling tool. And we can just do the same experiment if we want to. On the surface, we can mobilize microtubules, stain them, and then do halo tagged kinesin. And you can already appreciate the movement of the green color labeled kinesin on the microtubules that are quite obvious. And this work wouldn't be possible without my students, Aurimas, Nur, who did the software actually, and the optics. Aurimas and Meta and Justa, they were responsible for sample preparation and data analysis. And if you want to learn something more or get in touch, just don't hesitate to visit my webpage or just write me on X. And with this, I would like to thank for your attention, and I'm ready to answer your questions.